Hi everybody, welcome back to Neurobiology at Providence College. I'm Joe DeGiorgis. Today we're going to look at a paper by Ricardo Miledi titled Transmitter Release Induced by Injection of Calcium Ions into Nerve Terminals. So the title does a pretty good job of telling you what the paper's about and what he actually did in this particular experiment. So let's get started. Miledi, like Hodgkin and Huxley, used the giant axon from the squid. And we said the other day that the squid's body is shaped like an ice cream cone and that it's actually hollow inside. So water can enter and leave this ice cream cone shaped body, and that provides a jet propulsion mechanism that allows the squid to dart forward in this direction. If we take a pair of scissors and we cut along the bottom edge of the ice cream cone, we can then flatten the cone out, that is, open it up and flatten it out into a triangle like this. And we said that there are two neurons that come down from the brain of the squid. The organism is symmetrical. And that this axon, this neuron synapses with actually a series of giant axons. One is like this. There's another one here like this, and so on. And it's the same on the other side, of course, symmetrical. Something like that. So what Miledi did is he says in his paper that he used 3 different types of electrodes. He says 3 intracellular microelectrodes were generally used. One on the post-axon for monitoring synaptic potentials and thus transmitter release. So he has an electrode stuck into this giant axon to monitor action potentials, to see whether this neuron will fire if this neuron is fired. Synaptic transmission is a communication between 2 neurons, in this case, this upstream neuron and this downstream neuron, the squid giant axon. He says that 2 are in the presynaptic axon, which is this upstream cell. One electrode was filled with calcium chloride and placed within the synaptic region. So it was placed here, upstream, in this upstream cell, in the area of the synapse. And that contained calcium chloride. So the main point here is that it contains calcium. A second electrode was placed in this upstream neuron to cause this neuron to fire. That is, they would send electricity down this axon and cause it to liberate an action potential. So it's hooked up to a power supply that can be controlled, and you can send a signal down to this cell and get this cell to generate an action potential. And while this one contains calcium chloride, it can also monitor whether an action potential occurred in this upstream axon. So we have 3 electrodes, one here on the postsynaptic side, the downstream side, which is the squid giant axon. And we want to know whether firing this neuron will cause synaptic transmission to happen. That is, this neuron will release a neurotransmitter, and that neurotransmitter will stimulate this neuron, depolarize this neuron, and cause an action potential in this downstream nerve cell. So under normal circumstances, if we apply electricity to this electrode, this neuron will fire. You will detect the firing of that axon with this second electrode that contains calcium chloride, and then you'll have a downstream neuron, and if synaptic transmission happened, if neurotransmitter was released and that neurotransmitter was sensed by the downstream cell, then this neuron would fire and you would detect it in an oscilloscope here. I'm going to redraw the synapse schematically. So we have an upstream neuron, and if you look through an electron microscope at high magnification, the upstream neuron has synaptic vesicles that are in the presynaptic terminal. So this is the cell coming down from the brain. So this is upstream neuron. And then the downstream neuron, which is the series of giant axons, I'm only going to draw one, the one that's involved in the experiment, has the other side of the synaptic connection. That is, this is the downstream cell. And the idea is that if we stimulate this nerve cell with an electrode that's hooked up to a power supply, like a battery, then this cell will become depolarized and it will fire an electrical action potential. That is, the sodium and potassium channels will open and close and cause the electrical action potential. And if they had a electrode here, which they do, which Miledi does, and they hook that up to an oscilloscope, if this electrode causes this to fire, you will detect in the end over here an electrical action potential. When the electricity hits this part of the cell, the end of the cell, neurotransmitter is released by this presynaptic terminal. This is the presynaptic, or the upstream terminal, and this is the postsynaptic terminal. And we would have an electrode here, also hooked up to an oscilloscope. And if this neuron fires, you would see an electrical action potential in the downstream cell. So this shows that neurotransmission occurred, which means that the neurotransmitter was indeed released by following whether the postsynaptic neuron fires an electrical action potential. So under normal circumstances, if you stimulate this neuron and it induces an electrical action potential, you would pick that up in a recording by this downstream electrode, and you would see it on an oscilloscope that the neuron actually fired. And of course, if you put another electrode here and you measured this part on an oscilloscope, of course it would fire as well. You'd see that the neuron fired. This would happen prior to detecting this one. I mean, very, very fast in millisecond fashion, but obviously the action potential occurs here before it occurs down here. The action potential is a wave that propagates in only one direction down the axon in this direction. And if this neuron receives once the action potential reaches the terminal end of the cell, neurotransmitter is released, which diffuses through the synaptic cleft to receptors on the downstream neuron on the postsynaptic side of the synapse. And if there's enough neurotransmitter released and detected, then that would cause this downstream neuron to fire, and you would detect it with this downstream oscilloscope. So that's under normal circumstances. Miledi states in the very first sentence of his paper, as far as is known, all chemical synapses cease to transmit impulses if calcium ions are absent from the external medium. These experiments were done by Miledi previously, but what he showed is that if you have calcium, which does exist in the external medium, usually the squid's bathed in seawater, which contains calcium, and if you remove the calcium by adding something that binds to the calcium, like EDTA, which we'll talk about in more detail in the next paper, then the EDTA binds up the free calcium in the solution, and makes it ineffective. I mean, it sequesters the calcium. So there's no longer calcium in the external media. Now, if you stimulate the presynaptic neuron with this upstream electrode, the axon will fire, and that can be recorded by the downstream electrode here, and then detected by the oscilloscope. So you can stimulate the neuron, and the neuron still fires. It still propagates an electrical action potential, even in the absence of calcium. But now you don't detect a postsynaptic response. That is, you don't see an action potential in the downstream neuron. So somehow calcium is required for the release of the neurotransmitter and its detection by the downstream neuron. In this paper, Miledi is trying to provide further evidence that calcium is involved in the process of neurotransmitter release. If we go back to the title of Miledi's paper, transmitter release is induced by injection of calcium ions into the nerve terminal. So what he wanted to do was use this recording electrode to micro-inject calcium into the presynaptic terminal. So this was filled with calcium chloride. It's a needle, but the needle can record, and it also can inject. To do the experiment, he first added tetrodotoxin, TXT they call it, and that toxin is derived from pufferfish, and it binds to sodium channels and it makes them inactive. So it poisons this axon from firing an electrical action potential. So now, if you have the toxin in this cell and you stimulate with this electrode, you don't see a signal with this electrode, and you don't see a signal from this electrode. And, of course, you don't see a signal from this electrode, because there's no action potential produced by this stimulation because of the poisoning of the sodium channels. But now, if Miledi simply injects calcium into the presynaptic terminal, then he obtained a signal, a depolarization of the downstream cell, and saw an action potential propagated in this cell. And that's the finding of this experiment, that calcium injected into the presynaptic terminal of the synapse induces neurotransmitter release and the firing of the downstream neuron. So a short lecture today, but a really cool paper by Miledi that shows that calcium is involved in neurotransmitter release and in the process of synaptic transmission. See you next time.